Good morning, everybody. Today we will be doing chapters 3B and 4A, osmosis, etc. So I'm hoping some people can show up today. Good morning. How are you, Bella? You having a good morning? Good. Yeah, I have the chat open so I can see your responses. So be sure. Oh, I'm sorry to hear you were sick. Are you feeling better? Good. I'm glad to hear that. Okay, Bella, if we have anybody else join us, we'll go ahead and add them, but I'm going to go ahead and begin. I hope you have your chapter 3B and 4A lecture notes ready. So I'm going to go ahead and share my screen and we'll get started. Okay, this is chapter 3B, the working cell. Okay, we are going to be talking about some basic concepts in energy. So what is energy? It's the capacity to do work, the ability to do something. That's what energy is. There's different kinds of energy. The first is called potential energy. That is stored energy, energy that can be released to do something. So for example, you roll a rock to the top of the hill, wind up a spring, or stored in chemicals. Those are all energy that can be released to do something. Now, kinetic energy is energy in motion. That is if a rock is falling, a vehicle is moving, something is being done in the cell physically. So this is a figure showing the different types of energy. For example, the people at the top of these stairs have potential energy because they've added energy to themselves by climbing to the top. This is a person that has kinetic energy because they're falling, moving, and then anyone down in the water has much lower amounts of energy until they climb the ladder and add potential energy. Okay, chemical energy is a type of potential energy. It's stored energy that can be used to do work. The energy from the bonds between the atoms in a molecule can be converted into other forms of energy. And this is something you all should know. The more bonds a molecule has, the more energy it has. So for example, if you were in lab, you remember we created a molecule of glucose, and glucose had lots and lots of bonds, and all those bonds can be broken, reassembled, and that means glucose has a lot of energy. So to contrast that, we also did water and we did oxygen. Those are very small molecules. They have much less energy. So one of the review questions and test questions asked, does a small molecule like water or carbon dioxide have a lot of energy? And the answer is no. It's larger molecules with lots and lots of atoms that have more energy. The small molecules have very little energy in them. Okay, so carbohydrates, which is starches and sugars, fats, and gasoline even in our engine. It gets burned and can generate heat and movement. So plants and other producers, such as other photosynthetic prokaryotes is what I'm thinking of, convert inorganic molecules such as carbon dioxide and water, which have low potential energy. So look right there. These molecules do not have lots of energy in them, into glucose, which has high potential energy. And that's why they do it. So they have energy during the day. The sunlight is shining on them. How do we store that energy until we need it later? For example, at night, we put it into molecules. And then later at night, you can break those molecules apart, get the energy back, and stay alive, even though there's no sunlight. Now, we don't use sunlight directly. We steal those energy-bearing molecules from other living organisms. That's part of being a consumer. When these energy-storing molecules are broken down or burned inside the cell, energy is released and something is done. Okay, so this is a couple of good examples of potential energy stored in molecules. So for example, the octane molecule in gasoline plus oxygen, when you combust it in your car, heat energy is given off, but the most important thing is that it causes the vehicle to move. The byproducts or wastes are carbon dioxide and water. Now, food molecules such as glucose plus oxygen in our cells, very much like inside of a car, get burned, and they get burned in the presence of oxygen. Heat energy is also created, but the key is instead of kinetic energy by itself, movement, ATP is made, which can be used for a variety of things, including movement. But interestingly, the waste products are the same as in gasoline, carbon dioxide, and water. So these molecules have high energy, these have low energy. ATP and cellular work. ATP stands for adenosine triphosphate, T for phosphate, T for tri, P for phosphate. One adenosine molecule is bonded to 3 phosphate groups. Now, it's the last of those 3, the third one, that's the most important and carries the most energy. It's used to do all energy requiring jobs in the body. So if you need something that requires energy, you got to burn ATP to do it. One of the phosphates is removed, energy is released, and work is done. So if you break off one of the phosphates, that's what does the work. Now that molecule doesn't have as much energy as it did. So mechanical work, such as in your muscles, transport work, which is movement across a membrane against the gradient. We'll be talking about that later. And chemical work, converting reactants into products, sometimes takes energy. And all that energy is used in the form of ATP. Now, I know I'm going really quickly, especially for Bella, but when you rewatch this, or folks, when you're watching this later, be sure to pause, take detailed notes, not just fill in the blanks that are in your notes, but maybe take some notes off to the side of what I'm also saying. So this shows you mechanical work, muscles flexing. This shows you transport work. Notice that you're going from a place where there's very few solute molecules, which is particles dissolved in water, to where there's more. This is not the way they want to go. So you're pushing it against the gradient, against the way it wants to go. Things want to spread from high to low concentration. So when you're moving it the wrong way, it takes energy in the form of phosphate that comes from ATP. And then combining molecules together. For example, we take sugar molecules and combine it together into a starch molecule in our muscles and in our liver. That takes energy. So these are all energy requiring jobs in the body. Okay, what is selective permeability? Membranes of the cell are selectively permeable. Back in the day, they used to call it semi-permeable, which means partially permeable. Selectively is better because it means that the cell membrane gets to select which things move, which ones don't. They allow some substances to cross more easily than others. For example, water always moves easily. Gases always move, but large molecules and charged ions that we're going to be talking about later don't move as easily. They block passage of some substances altogether. Without this selectivity, the cell could not survive. So that's what allows cells to be alive. Now, it's easier to do it inside of a body because the cells are in a nice, stable environment. But if you're a single-celled organism out in a swamp somewhere or in the water, this selectivity is super important to allow you to survive. Okay, these are some membrane protein functions. I don't believe it's going to be on the test, but I'm going to go through it briefly because I may be asking you about some of the functions. So the first one is attachment to the cytoskeleton extracellular matrix. That means that these proteins, which are all the purple things here, can be connected to fibers that are part of bone or cartilage that allows the cell to stick to those structures. In B, cell signaling, a molecule like a hormone can fit into this little groove on it, causing what they're showing here, a chemical reaction inside the cell. So what does that mean? It means that these molecules, by sticking to the outside, are able to control the inside of the cell's cellular machinery and metabolism just by sticking to the outside. In C, this is enzymatic activity. Notice that one molecule comes into a protein that has the same shape as it. It's converted into a circle. The circle then fits into a separate one, which also has the same shape, and it gets broken in half. What does this mean? Some proteins are enzymes. Enzymes break molecules apart. They can assemble them together, and they can change them into different molecules. You see the square turns into a circle and the circle in half. In D, transport, we saw earlier the movement of molecules through the membrane. Some proteins act as channels that allow things to come through. In E, intercellular joining, this is physically the two cells being stuck together. Now, this is different from being connected to something like bone or cartilage. Here, the cells are directly connected to each other, gluing them together physically. And last but not least, cell-to-cell recognition. This is how cells are able to tell whether or not the cell on the bottom belongs to part of your body or is a foreign invader. Notice that it fits exactly into a little cutout on the upper one saying, hey, this is part of me and not a foreign invader. But if the molecule sticking up was from a foreign invader and it was recognized, this would be a white blood cell and then would destroy it. That's part of your immune system. Okay, so getting through the membrane, diffusion. Now, what does diffusion mean? It means particles spreading out from where there's lots of them to where there's less. Well, look at the picture on the bottom. You see lots of material. And this is a picture from your text, by the way. See lots of particles here, and this is the membrane. As they spread out through the membrane, you can see they come to what is called equilibrium. That means there's equal amounts on both sides. So particles want to spread out and expand out. In the same way that a crowded elevator, when you open the door, you don't have to push people out. They will get out on their own. Or if you have a crowded room and you allow people to go into a larger room, they will generally spread out from a crowded place to a less crowded place because they want more space. And that's what particles do. So diffusion is the movement of any particle from an area of higher concentration to an area of lower concentration. Relative just means more or less compared to the other side. So notice that here there's more than this side because there's a lot and none. And here there's equal amounts. So there isn't a difference in the concentration. That would mean there's no concentration gradient. Here there's a high gradient because the difference between here and here is the gradient. The concentration gradient refers to how much difference there is between the two concentrations. And notice in the middle, there's still a gradient, even though it's becoming less. Less of a gradient as particles move. And then here, they're at equilibrium, equal amounts on both sides. Now, what's important to understand on this last picture is that even though the amounts on each side aren't going to change, sometimes particles go one way while another one goes the opposite way. So just because it comes to equilibrium doesn't mean stuff stops moving. It just means the amounts no longer change because every time one goes one way, another one goes the opposite way. In this case, almost all of them are going down, mostly down here, and equal amounts up and down there. The net movement refers to the overall movement of molecules, not to individual molecules. So in this case, on the far left, the net movement is from top to bottom because there's almost nothing coming back. Here, the net movement is still from top to bottom. But the net movement here is zero, because every time one goes down, another one goes up because they're in equal amounts. So even if net movement is zero, that means the change is zero, doesn't mean nothing is moving. So please be aware of that for the test. This process is what moves carbon dioxide into the air of your lungs and eventually out of you. And what moves O2 out of the air of your lungs and into your blood. So it's going from out, when it says out of your lungs, that means the air of your lungs, into the blood of your lungs and into your body. And this moves the CO2 from the blood into the air of your lungs. So when we say lungs, we mean the air of your lungs, not the blood, okay? Diffusion is passive. It doesn't take any ATP energy. The particles are moving, so they naturally just spread out as a result of their movement. Okay, so you can see this is another figure showing how it works. Same thing, high concentration here, very little here, and it spreads out to equilibrium. But this figure shows a little bit more. This is not in your text. What this one shows is that each one moves relative to its own concentration. So blue is spreading out from left to right, but look at red is also spreading out from right to left. So they're both moving in opposite directions. The net movement for blue is to the right, net movement for red is to the left, and they're independent of each other. They don't interact in any way. So one can be moving one way and one can be using the other. So this is how oxygen is coming in. Imagine oxygen is blue coming in while carbon dioxide is leaving in your lungs at the exact same time in opposite directions. Okay, osmosis. I don't know if you guys have seen this in your text, probably haven't, but you can see here what we have is a semi-permeable membrane. What this means is that the water molecules, which are shown in pink, can move, but these big purple molecules cannot. So what's going on here? Let's take a look at the picture before we do the notes. Now, these particles stick really well to water. They like to stick to water. So what happens is, because there's so much more on the right, when a water molecule crosses over, it gets stuck on these purple molecules and can't come back. Now, a little bit comes back, but mostly it goes across and gets stuck on these lots of what are called solute particles, or anything dissolved in the water. Now, since more are going to the right than are going to the left, water accumulates here. Now, even though there's overall more water, you can see that the amount of water compared to the amount of solute is even on both sides, the spaces between them. So if you allow osmosis to happen, when there's a whole bunch more solute or dissolved material, you'll see the water will go to where there's more salt or dissolved material, okay? So that's osmosis. So what is the definition? The definition is the movement of water molecules from an area of higher concentration of water to an area of lower concentration of water. So if you notice the picture on the left side, there's more water than on the right, because on the right, there's more stuff, there's more solid. So if there's more solid, there's less water. So the water is moving from left to right to equalize how much water there is compared to how much solid there is. Selectively permeable, we mentioned this, it means that molecules can pass through. Always water can move, and most cannot without the help of channel proteins. So if there's no special tunnel to allow these molecules to move, they can't move, but water does. In biology, the solvent is always water. That means the liquid portion is always water, and the solute, which is the dissolved portion, is anything dissolved in water. So that can be salt or protein or oxygen or anything. Now, in the case of my analogy, I'm going to be talking about the solid as being a salt. But remember, it can be anything that's dissolved in water. The key to remembering this is water is always attracted to solids or salts. So notice that the water is attracted up here from left to right because there's more solid or salt on the right than on the left. And the solute can't get over here. Now, if the solute could, it would just go to equilibrium and the amounts wouldn't change. But because these purple circles are stuck there, the water goes across and gets stuck as well. And so the volume increases. Okay, so in osmosis, water always moves from an area of higher concentration of water to one of lower concentration. In this system, the solute cannot pass through the selectively permeable membrane. And this image and this figure notes are directly from your text if you want to take a look at it. Okay, more about osmosis. If a cell has more solute or salt in it, then it's said to be hypertonic to its environment. Now, I want to have you guys understand what hypertonic means. It just means saltier. It doesn't tell you how much salt. It just means it's saltier than a different place. So hypertonic in this case means the cell is saltier than its environment. It just means saltier. It's not an exact amount. It's just a comparison. More salty, more stuff dissolved. As we saw in the previous figure, the right side was hypertonic, having more stuff in it than the left side. And that's why stuff moved towards it. If a cell has less solid, the cell is hypotonic. So that term hypotonic just means less salty or less stuff dissolved in it. So it's just a comparison. So you could say the cell is hypertonic, the environment is hypotonic, or vice versa. It just means where there's more or less salt or other dissolved solids. If there's equal amounts, like in our body between our blood and our cells, that's what's called isotonic. Iso means the same. And so in the case of our body, the amount of salt and total dissolved solute in our blood is equal to our cells, so water neither moves in or out. Okay, so what effect does it have on cells? If the environment, the water around a cell, now let's talk about an individual cell like a paramecium or a single-celled organism in water. If the water is really salty, then the water will move out of the cell and into the environment, and it will shrivel. It will shrivel up because the water is leaving the cell, because water is always attracted to where there's more salt. If the environment is hypotonic, so let me give you guys another key thing. It's that cells always have a certain amount of salt. It turns out the amount of salt in our cells is equal to the amount of salt in the earliest oceans on our planet, which is kind of an interesting coincidence. I don't think it's a coincidence, but hey, it's similar. But if you put a cell which has a certain amount of salt in it, which I told you was less salty than the ocean now, but saltier than fresh water, than rainwater. Cells have salt. So if you put it in pure water, which has no salt, the water will come in towards the cell because that's where there's more salt or solid. So the cell will tend to swell up and burst if it doesn't have a cell wall. Now, our cells don't have a cell wall. So why don't they burst? Well, remember, we don't put our cells in pure water. We put our cells in our environment of our plasma, which has the same salt as our cells. And if we drink pure water, it gets dissolved and dispersed throughout our salty blood, also not causing our cells to swell up and pop. But if you did put one of our red blood cells in pure water, because it has salt in there, the water will come in and it will burst because our cells have no cell wall. If a cell is isotonic, for example, our blood and our cells, it will maintain its shape. Now, quick question. If no net movement of water is happening, is there any movement of water at all? What do you think, Bella? If there's no difference in the way they're moving, it doesn't mean that there's no movement. What do you think, Bella? Not sure? Okay. The answer is yes, water molecules are still moving, but for every one that goes in, another one goes out. And in an isotonic environment, they'd be moving in equal amounts. Okay, now I'm going to be talking about red blood cells and lettuce leaves, and this is likely to be a essay question on the upcoming test, so you do need to take detailed notes on it. So we talked about isotonic, hypertonic, and hypotonic. We're going to talk about how plant leaves are different than red blood cells. Okay, so let's talk about, in the first case, we're going to start right in the middle. We're going to start in the middle here. I know it's not normally where we start, but in an isotonic solution, the red blood cells, which have this shape, remain this shape because if you look at the cell itself, as water comes in, so does it leave, because there's equal amounts of salt on both sides. And the same is the case for the lettuce leaf or plant cell. Where it gets different, though, is on the other sides. Now, if you look at red blood cells in a really salty, for example, if you put red blood cells in the ocean, let's say you bleed into the ocean, this is what your red blood cells would look like after a short amount of time, because the water is leaving and going to where there's more salt, so it shrivels up. These are shriveled up, which look like dried out red blood cells. Now, the plant cell also shrivels up, but notice that the cell wall, the black edge, does not change shape. But this is what happens when plant cells wilt. This bubble in here, which is full of water, the water leaves, the cell shrivels up, and now the cell wall isn't as rigid as it was before, and you see a wilted plant leaf. Instead of being up normally, it's wilted and draping downward. Let's go to the far right. In a hypotonic solution, remember, hypotonic means less salt. So in this case, it's saltier inside of the cell than out in both situations. So in this case, the water comes in. Now, because red blood cells, no animal cells do, have a cell wall, it can't resist the water coming in. And you can see right up here, they swell up and they burst and they let all of their contents out. This is a medical trick for getting what's on the inside of our cells to the outside. So you can do what is called hematocrit to check how much hemoglobin, or that red colored pigment that carries oxygen in our blood, how much our cells have. So you put it in pure water, they all burst open, you move the outside of the cell down to the bottom using a centrifuge, and then you can check how red the solution is, tells you how much hemoglobin there was in the blood. Okay, now in a plant cell, water is also moving in, but why doesn't it burst? Well, what do plant cells have that we don't? They have a cell wall. Now, the reason why it doesn't burst is because the cell wall prevents that from happening. So the force of the water coming in is given back by the strength of the cell wall. And this is kind of how a car tire works. Now, you have your car tire inflated to a certain amount, let's say 30 psi. When you hit a speed bump going fairly quickly, the pressure in the tire goes up over 100 psi. Well, why doesn't it rupture? Well, because the fibers in the walls of the tire prevent it from exploding in the same way that the cell wall keeps a plant cell from exploding when it's in pure water. Okay, so I'm going to ask you to tell me about these different environments and why are our cells, our red blood cells, different from those of plants. Well, remember, it's the cell wall, but the movement of water is the same in each of these cases. And you can see it's pointing the direction of water, and you just need to tell me all about this. Okay, it is an essay on an upcoming test. Okay, what is turgor pressure? Well, the turgor pressure, if you remember me showing you the bubble, the blue bubble in the center of the plant cell, the plant keeps water in there because it needs lots of water to do photosynthesis. When we get to chapter, well, used to be chapter 7, but the chapter on photosynthesis, we'll be talking about what exactly do plants use water for in photosynthesis. But here's a happy plant and here's an unhappy plant. So in most cases, this plant becomes like this plant because you didn't water enough. So where does the water go? Well, the plant is actually using the water to make food. So it uses up the water. That's why you have to constantly water plants. But the question I have for you, and since there's only one of you and Bella is on chat, is how could this plant become like this plant by being watered? This is a kind of not a trick question, but asking, do you remember the different types of water that can go around a cell? Well, I'm not expecting Bella to get it by herself, so I will give you the answer. If I took this plant and I watered it with salt water, it would look like this. And the question is why? Why would it look like this when I watered it with salt water? Well, remember, water always goes to where there's more salt. And since there's more salt in the soil than in the plant would actually shrivel up and die because the water would be sucked out of the plant into the soil, getting to where there's more salt. That's why you can't water plants with salt water. You need pure water to do that, or tap water, which is not pure, but good enough for the plants. And this is another example of that is why you can't drink salt water, why you can't drink ocean water. It's got water in it, but it's got so much salt that if you put it in your stomach, the water would leave your body and go into your stomach, which is the opposite of what you want. You want the water to go into your body. When you drink pure water, since your blood is saltier than what you've consumed, it goes out. The water goes into you. But if you drank salt water, the water would leave your body and go into your stomach, making you thirstier. That's why you can't drink salt water. Okay? Okay, chapter 4a, enzymes. So we're going to be talking about enzymes here. Now, all molecules in our body are constantly being built up, which is called anabolism. For example, anabolic steroids. We're going to talk about that a little bit, or broken down through a process called catabolism. So anabolism means build a molecule. Catabolism means break a molecule. Normally, anabolism takes energy. Normally, catabolism releases energy. Now, if you add up all of the buildup processes and all the breakdown processes, you get to what is called metabolism, which most people think of how fast do you burn food. But metabolism is all the chemical reactions in our body, not just how quickly you burn food. So just to let you know. Most chemical reactions require an input of energy before they will start. This is called activation energy. For example, if you want paper to burn, you have to add heat. If you want gasoline to explode, you have to add a spark. That is what is called activation energy. You have to add energy to get a whole bunch more out. And the same is true for the chemical reactions in our body. Now, this is a figure showing how enzymes work. Now, normally in purple, you would need to add this amount of energy going up is how much you have to add to make it happen. And the amount downward is how much energy you get back. So in the purple line, you have to add a whole bunch of energy to get some back. But when you put an enzyme in there, notice that the amount that you have to add is much less. So normally, if you want to burn paper, you have to get it really, really, really hot. But those same molecules in a slightly different configuration will burn in our bodies at this much lower temperature if there's an enzyme present. So what it's saying is enzymes lower the activation energy to take the reactants to the products. You only have to add this much rather than this much. And that allows chemical reactions that are similar to burning to happen in our body at body temperature rather than getting it up to 400 or 500 degrees, which is normally what you'd need to do to burn something without an enzyme. Okay, if you want chemical processes to speed up, you can make things warmer. Well, that's one of the reasons why our bodies are warm, is because it makes chemical reactions happen quickly. But can you continue to add temperature to our bodies indefinitely? No. Increasing temperature inside of a living thing can cause fever and death. As you guys know, we sit at about 98.6, which is 37 degrees Celsius. If you go up above 100, 101, 102, then people start getting in trouble. And that's only a little teeny tiny difference between our normal and a fever. That's only a few degrees. What that means is that our body temperature is right up near the edge of being in trouble. You can also speed up chemical reactions using a catalyst. So instead of making it hotter and hotter to make things go faster, you can add what is called a catalyst. Now we have catalysts in our cars. And I've been reading on the news that people have been stealing these things in cars called catalytic converters. And what do catalytic converters do? Well, when you burn gasoline, as you saw earlier in the lecture, waste products come out. But what you didn't see is what also comes out without a catalytic converter is a bunch of unburned gasoline, which would turn into really nasty smog and pollution. So when it goes through the catalytic converter, what it does is it adds extra oxygen. The automobile adds extra oxygen to that waste. And then when it goes over the catalytic converter, it finishes burning the unburned gasoline in that catalytic converter. So what comes out is much more clean. But the reason why they call it a catalytic converter is because you don't have to add energy to that, and it doesn't have to be really, really, really hot. It only has to be as hot as the exhaust, which is actually a lot cooler than the inside of a temperature, the explosions in your car. So the catalytic converter is there to finish the burning process. That's why they call it a catalytic converter. And people steal them because these contain rare metals that do this job, but that you could sell. And so if you steal a catalytic converter, you can sell it for money, I guess. It seems sad, but such is life. A catalyst is any molecule that speeds up a chemical reaction without being changed itself. For example, the palladium and platinum inside of a catalytic converter speeds up the reaction of oxygen with unburned gasoline coming out of your engine to allow it to be cleaner. Okay, a protein molecule that acts as a catalyst is called an enzyme. So all enzymes are made out of protein. So what it does is it causes chemical reaction to happen at a lower temperature and doesn't get changed itself. That is the nature of a catalyst. Most of the DNA in the nucleus of all living things is used to make enzymes. So that's why we have so much DNA, is because we need lots of different enzymes to do lots of chemical reactions in our body. It turns out that you need a different enzyme for every different chemical reaction that happens in your body, and there's lots of different ones that happen. Enzymes build all the molecules that are in our cells and break down all the molecules in our food. So anything that's going to happen chemically inside of our body requires an enzyme. And you have to have a specific enzyme for every different chemical reaction. So notice this is one I want you to write all out. The most important biological molecule, excuse me, on the planet is enzymes. They are responsible for all biological chemical reactions. DNA gets all the press, is a lot more sexy molecule, but it's proteins and enzymes that do most of the work inside the body. Okay. How do enzymes work? Okay. Well, each enzyme is specifically designed to function with a molecule that is called a substrate. So the substrate is a molecule that's going to interact with the enzyme. The place that the substrate fits into on the enzyme is called the active site or binding site. That's the place where the substrate is going to bind. So if this is the enzyme and this is the substrate, the spot where it fits is called the active site or binding site. If the active site changes shape, the enzyme can no longer work because it doesn't have the right shape for the substrate. If it has a different shape like this, it can't fit and the enzyme doesn't work. So here's an example from your book. So here's the substrate, the molecule is going to be changed, and see that the enzyme has an active site that matches the shape. So when they come together, it is what is called the enzyme substrate complex. They're all connected together. Now, with or without energy, usually with energy, the enzyme product complex. Notice that this is one molecule made up of two connected pieces. But here the enzyme product complex is two separate pieces that are now connected. The bond between these two green molecules has been broken. Now we have a blue and a yellow. And what happens is the blue and yellow leave. What happens after they leave? Another green one comes in and goes through the same process. So this is directly from your book. Induced fit model is an adjustment to the lock and key model and explains how enzymes and substrates undergo dynamic modifications during the transition state. Anyway, this is from the book. It basically just means that they come together, the molecules get broken apart, and then they leave and it repeats. All right, so how do we name enzymes? Enzymes are almost always named for what they do. You can tell what an enzyme does just by looking at its name. The name of an enzyme almost always ends in the suffix ase. Now, what is the suffix for sugars? You guys have heard of sugars: glucose, fructose, sucrose. What is the three letters at the end of all of those? Well, it's OSE. OSE or OS means sugar. ASE means enzyme. Okay, so when you see a word that ends in ASE in biology, that means an enzyme. Not all enzymes have the ASE ending, but most do. So, for example, the enzyme that breaks down the starch, which is a bunch of sugars hooked together, is called amylose, and you call it amylase. So you see the amylase connects to amylose to break the starch into sugars. The enzyme that synthesizes glycogen is called glycogen synthetase. It tells you exactly what it does. An enzyme that synthesizes glycogen. This is how we make starch in our liver from the sugar in our food that comes in through our blood. The enzyme that helps DNA form a polymer is called DNA polymerase. So this is how we grow our DNA longer when we need to make more. What does lactose dehydrogenase do? Well, just look at the end. It's an enzyme that dehydrates, which means takes water out of lactose, which is milk sugar. So you can tell what an enzyme does by looking at its name. Okay, so what effect does the environment have on enzymes? Well, first of all, let's take a look at this figure. What it's showing you is on the x-axis, which is left to right, this is temperature in degrees Celsius. Scientists use degrees Celsius. So human body temperature is 37 degrees Celsius or 98.6. Right there, that's 98.6, just shown with a different scale. Now on this scale, the up and down or y-axis, it shows you how fast the enzyme is working. So as we go from left to right, what we see is the speed is increasing and increasing up to a maximum. So this is how fast it's moving. It likes to move at about 60,000, and that's at body temperature. But look what happens to the enzyme as you get it past ideal. This is fever here. This is when you have a fever. Notice your enzymes are slowing down. Why are they slowing down? Because you're destroying with the extra heat. This is why you die from heat stroke. This is why you die from a fever. Another thing this figure shows is an egg being cooked. Well, a raw egg, the white is clear. A cooked egg, the white is white. And why is that? Because the shape of the protein has changed. So let's do the notes. This figure shows enzyme activity increases with increasing temperature. This is the rise in the activity as we move from left to right. If reactions are too cold, reactions happen too slowly. So if we go to the left, you can see you're not doing enough with the enzyme because it's moving too slowly. We want to be way up here. So we have to be around body temperature. So one of the reasons that people die due to hypothermia or being too cold is their enzymes are moving too slowly to make the energy they need to stay alive. If the temperature goes too high, the enzyme is permanently damaged. The word that you need to use is called denatured and cannot function. So as you move to the right, the enzymes start to be denatured or destroyed and have to be broken down and made into new ones. And this is one of the reasons why you're so tired after a fever, is because a lot of the enzymes responsible for you having energy have been destroyed by the high temperature. So why does our body do it? It's not a mistake. It does that to allow our white blood cells to move more quickly, and it also destroys a lot of bacterial and viral cells by that increased temperature. We have a lot of extra enzymes that we can draw upon after a fever, but if we destroy too many of the fever goes too high or too long, we die because too many of the enzymes that give us energy were destroyed and we don't have enough left over to stay alive. That's one of the reasons why you want to get a fever down if it goes up to 105 or so, because that's where the enzymes really start to get destroyed quickly. Now the question is, if this is an egg and the white is made out of protein, how can we uncook this egg? Well, it's kind of a trick question. If you remember, I said that the enzymes, which are made out of protein, have to be broken down and reassembled after you're done with the fever. The only way to uncook an egg is to chop it into small bits and feed it to a chicken. I know that sounds kind of disgusting, but what does the chicken do? It breaks all the particles of the egg down, all the proteins into amino acids, and builds a brand new egg in the same way that we break down the destroyed or denatured enzymes, which are made out of protein, and make new ones. So that's the way to uncook an egg. Okay, this is another figure that shows not only is it temperature, but pH, how acid or base the environment is. It's very specific for pH. Now, what I want you to see on this is that each of these enzymes, notice only one of them has the ASC ending, these two don't. Each of them has this pink zone, which is its happy place for how much acid or base there is. Pepsin likes it in acid, this is in your stomach. Amylase likes it best in your mouth. And trypsin, found in your small intestine, likes it slightly alkaline or basic. If the enzyme is too acid or too basic, then they will be denatured. Let's take a look at amylase. If we go too much to the acid side, notice its activity, the enzyme activity drops off because it starts being denatured. But the same is also true to the right. So anything outside of its pink comfort zone, it's going to stop working as well and become denatured. So what it's showing is that each of these enzymes, the enzyme found in your stomach, remember your stomach has acid in it, right? So pepsin actually likes the acid. It breaks down protein in your stomach. But if it gets too neutral or even more acidic, it will be destroyed or denatured. The same is true for the amylase in your mouth and the trypsin found in your small intestine. If you get it too far away from ideal, it will stop working because it becomes denatured. Okay, so these pictures may be on the test, and I may be asking you, what are these figures showing? And that's what's happening. So when amylase is in your mouth, you start digesting starch in your mouth, then it goes into your stomach. Pepsin starts breaking down the protein in your stomach. It likes the acid. Then you go all the way back to the small intestine, trypsin. And the fact that it switches all the way from neutral to acid and back, what that does is it kills a lot of bacteria and viruses in our food. All of our food has bacteria and viruses in it, and the acid in your stomach helps destroy that. Another function of the acid is to unfold or denature the proteins in your food, making the enzymes there better able to access it. Please note the acids don't digest the food. They just denature the proteins, unfolding them, allowing the enzymes to get in there and break them up into the parts that we can absorb. Okay, enzyme inhibitors. What does an inhibitor do? Well, to inhibit someone or to inhibit yourself means to stop doing something. What it means is they can slow down or stop a metabolic reaction, and this can be good or bad. Some can bind to the active site in the place where the substrate would go and act as substrate imposters, thereby blocking the enzyme from working. This is called competitive inhibition because this molecule, which is not the substrate, is competing with the substrate to get into the spot where it fits. So it's called competitive inhibition because they're both competing with each other to find that active site. Now, here's a good example of it. Well, this molecule, this figure should have been after the next one. Okay, so we're a little bit out of order, my mistake. Now, this is another way that you can turn off an enzyme. So notice that we have the correct shape active site for the substrate, but in this second kind of inhibition, the inhibitor doesn't fit into the active site. It fits into what is called the remote site or allosteric site. So notice when the inhibitor fits in, it causes the active site to change shape and the substrate can't fit. But some activators, instead of inhibitors, actually make the active site change to the correct shape. I'm not going to be asking you about this one. I'm going to be asking you about this one primarily. And that is you have the proper shape of the active site to fit with the substrate. But when the inhibitor comes in, now it won't. This is a way to turn off an enzyme. This can also be either good or bad. So this figure should have come after the next slide. Sorry about that. Okay, this is another example of inhibition. So notice that we have the correct shape of the active site for the substrate, and when the inhibitor comes in, it gets smaller, changes shape, can't fit. It binds to a remote site, or sometimes called the allosteric site. And sometimes, in some cases, it's called feedback regulation, and I'll show you what I mean by feedback regulation. Sometimes you want to turn off enzymes because you don't want them going 24 hours a day. This is called allosteric competition or non-competitive inhibition. So allosteric inhibition or non-competitive inhibition are both correct answers. So be looking for that on the test. Now, how does this work? Now, how does feedback regulation work? Well, let me show you. In this case, this is directly from your book, by the way. So if we're looking at enzyme 1, turning this purple molecule, the green into purple, then purple fits into enzyme 2, turning it into blue, and blue fits into enzyme 3. Well, if you have this end product and you have enough of it, the body only wants a certain amount. More is not always better. Just the right amount is what is better. So sometimes if you make enough of this end product, what it does is it comes in and prevents the green molecule from turning into the purple molecules. Notice that the shape of the active site has changed. Now you can't take the substrate and head it down the line. Now, why would the body do this? Well, you have plenty of the end product. What's a good example that you have in your house? An ice maker. How does an ice maker work? Well, if you take ice out of your ice maker, that little metal arm drops down, and that tells the ice maker it's time to make more ice. So the ice maker makes more ice and dumps it into the bin, and the little arm checks how much ice is there. If this bin is full and the arm comes down and it can't go very far down, it shuts off the ice machine. Doesn't mean it's broken. It just means you've got enough ice. When you take the ice out, the arm drops, and that turns the machine on again. And that works kind of like what's happening here. When you use up the product and you don't have enough, the whole system turns on again. It's because this end product is no longer there to inhibit the enzyme. And it starts up again, starting to make this product in the same way that ice is made when you draw ice out of your ice machine. This is called feedback inhibition because you're going from the end back to the beginning and inhibiting it. Does that make sense, guys? The end product feeds back from the end to the beginning and shuts off the thing that would start the process. In the same way, the ice turns off the machine that built it. So it's called feedback inhibition because it feeds back from the end to the beginning and turns off the thing that would make it temporarily until you need more. Okay, that is it for today. Bella, you're the only one here. Do you have any questions about what we just did? Okay, well, we do have lab. I will be in lab next week. We will be doing a lab, so please come to lab. I don't know if you guys got my, I'm going to be out. I'm going to be out. Let's see, where was it? It was on Monday. I don't, yes, it's going to be in T10. It's going to be a T10 because remember we moved from T14 to T10. And we will be in there on Monday. I don't remember if I told you all in lab that I wasn't going to be in class on Monday. If I didn't, that was my mistake. I should have. But I did email you in the morning. And if you ended up on campus and saw the note, I'm sorry that you didn't get the information sooner. I meant to tell you the previous Monday that I was going to be out on Monday. But if you didn't get it or I forgot to tell you, I'm sorry. But we will be in class on Monday. We will be doing a lab. Please be sure to bring your lab report with you. That was the atoms and molecules lab report, and turn that in on paper. So you've got now 2 weeks to do it. And then we'll be doing a lab that will be due the following week. So on Monday, we'll be doing a lab that will be due the following Monday. Okay, well, I do appreciate Bella, you showing up, and everybody else watching. Please use this to fill in your notes, take detailed notes. And when I see you on Monday, let's see, let me just double check this really quick. Do you guys have you do not have a test this week. Okay, that's good. So because you don't have a test, so what we'll do on Mondays, we'll also take some time to review for the test. And then we will also spend some time reviewing in lecture for anybody who can show up. But I know since many of you have difficulty coming to these lectures, we'll try to do some review on Monday for the test next week. Okay, hey folks, enjoy the rest of your week and work on those review questions, finish up that lab report from last week, and hope to see you on Monday. Have a good day, guys. Bye now.